liver and bile. Pancreas, liver, which is produced by the bile, which is produced by the gallbladder. Okay, clear? Am I fast? Yes? Please cope up with this because we cannot finish the topics in the required time. Okay? So I'm just listing out the important functions and just explaining you the concepts. If you have any doubts, please raise your questions. Okay, clear? So now, pancreas is one of the accessory organ. Okay, it is indirectly helping in the digestion, but the food chyme or the bolus is not contacting with the pancreatic structures directly. Only the secretions helping in the digestion. In the same way, the pancreatic enzymes are the main source which is involved in the digestion of carbohydrates and the proteins. And next, we have to talk about the lipid digestion, which is majorly mediated by the bile and its liver and its secretions. Okay, so this is also a accessory digestive organ. So if you talk about the single cell of the liver, it is called as hepatocyte. That is hexagonal shape structure. Okay, so you term it as the hepatocyte. So the central part of hepatocyte, we call it as central vein. In all the corners, you have a triad. Okay, so this triad includes the pathway for bile, portal vein and hepatic artery, which constitutes the triad. So here you have another hepatocyte and here you have another hepatocyte. So this is how the functional unit of liver is giving the cellular organization of the liver. Okay, and next we will move on to the physiological aspects of the liver. The main secretion of the liver is, you have the liver and below that you have, what is the structure? Gallbladder. So the liver has right and the left bile duct. Okay, whatever bile is being synthesized in the liver, it is stored in the gallbladder. Gallbladder is not synthesizing the bile. It is just the storage bag. That is why in case of gallstones, even if you remove the gallbladder, the person is not having any abnormality in the digestion. It is not disturbing the digestive function because gallbladder is just only the storage bag. Okay, so from the liver, the right and the left bile duct is coming out, and this forms the common hepatic duct. From the gallbladder, you will get the duct that you call as cystic duct. Okay, and now the duct from the bile as well as from the gallbladder, it is combining together and it forms the common bile duct. Okay, so this common bile duct and the pancreas, which is going to give the major pancreatic duct. Okay, both are now going to join at the region which you call as the second part of the duodenum. And same here, the bile duct is also going to join. And this part where the bile duct, common bile duct and the major pancreatic duct, which is joining, it is called as of Oddi. It means it is having a sphincter muscle over there to prevent the reverse flow of the bile secreted from the liver as well as the pancreatic juice. Okay, so that is in the second part of the duodenum. Once when the contents digestive function in the duodenum. Okay, so this is about the liver, and apart from this, the other significant functions of the liver, we will just list out the significant functions. The other significant function is formation and secretion of bile. Second one, liver plays a major role in detoxification process. Which of the cellular organelle is rich in liver to perform this detoxification process? Anyone? Which cellular organelle of the liver is performing detoxification? Just listen the question. Peroxisome. Yes. Peroxisome, when you are studying it under the cellular organelles, it is the place where the oxidative damage is prevented. This is the place where the catabolism of lipids is taking place. Beta oxidation of fatty acid is taking place. And we have also seen this peroxisome is rich in liver to perform the detoxification function. Why is because of the peroxisome. Okay, so liver plays a major role in detoxification function. And then third one is, it is the also having storage function. What are all the things stored in the liver? Glycogen, it is the storage form of glucose, and it is also storing various vitamins, mainly vitamin A and B12. Okay, and it is also storing iron in the form of, what is the storage form of iron? Ferritin. From where the iron is coming out? From the fate of hemoglobin. After 120 days, the RBC is lysed and it is releasing hemoglobin, which is further releasing the iron. It is again reutilized for RBC synthesis, and the excess will be converted into transferrin. The transferrin is the storage form of iron. Okay, it is also happening in the liver. And apart from this, liver is also having endocrine function. What is the endocrine function of the liver? It is the place where the vitamin D activation is taking place. Activation of vitamin D, 1,25-dihydroxycalciferol. Calciferol conversion, it is taking place in the liver, and also it releases the hormone called as insulin growth factor, which is also called as somatomedin C. These are some important endocrine functions mediated by the liver. And the major one is it is the main storage reservoir for the blood. Okay, it reserves nearly 650 ml of the total blood volume. Okay, so this is the storage reservoir of the blood, and it is also having hemopoietic function. In the erythropoiesis, in the hemopoiesis, we have seen embryonic state during the fetal life, then hepatic state during the second trimester, and then only it is going to the myeloid stage, which is being taken by the red bone marrow, and on emergency situations by the yellow bone marrow. So liver is the one which is involved in the hemopoiesis process. Okay, and finally in immunity, which is mediated by which cell? Which cell in the liver is doing the role of immune function? Kupffer cells, which are the modified form of macrophages. Okay, so these are the some peculiar functions of the liver. And next we will move on to the concept of enterohepatic circulation of bile. Okay, so we have seen that bile is secreted in the liver, okay, and then stored in the gallbladder, and it is entering into the duodenum for the digestion of lipids mainly. Here the bile will undergo the formation of chylomicrons and then vesicles, which is involved in the digestion process. All those in detail you will study in biochemistry. Okay, the formation of chylomicrons, vesicles, where the conversion of VLDL, HDL and other things, all these things will take place. Okay, so after all those process, it is entering into the intestine. Everything is done, and whatever bile secreted from the liver is nearly 100%. Okay, after doing all the functions, then the conversion, conjugation, bilirubin, everything will be done. Later on, before excretion, only the fecal matter is excreted out, and only the 5% of bile will be excreted. And what happens to the remaining 95% of the bile? It will again go back to the liver. This is called as enterohepatic circulation of the bile. So liver secretes nearly 100% of the bile, which is going into the enteric system, doing all its function, and during excretion, along with the fecal matter, only 5% of the bile is excreted. The remaining 95% is going back to the liver. So from the hepatic region to the enteric region, back to the hepatic region, you call it as enterohepatic circulation of the bile. So every time the liver should synthesize only the deficient 5% of the bile, because 95% is coming back to the liver. Okay, clear? So this is what is about the enterohepatic circulation of the liver. If there is a damage in the liver, then what kind of abnormality in the digestion you will encounter? Liver damage, cause may be of anything. Lipids. Lipid digestion will not happen properly, no proper bile secretion, and liver is damaged, so it will not get the bile back from the enterohepatic circulation. So you will encounter all those things. That is what you are terming as the hepatic jaundice. Okay, clear? So next, we are going to see the small intestine.